what I'll be talking about is effectively continuing what the previous speaker talked about on targeting senescent cells. And also, this was already introduced by Marco, so the benefit of being a third in the session is I don't need to introduce senescence anymore. I always say, like, we need to target aging because the 2/3 of the years we gained over the last generation were bad years. And as I'll show you, in mice, at least targeting senescent cells is one of the few things, aside from dietary restriction and compounds that try to mimic dietary restriction, that actually can extend the healthy lifespan of mice. So that was my introduction, what I usually use 10 minutes for, so that was it. So that brings me to this slide. In my lab, I think these are the three keywords. So senescence, and I think that's a cause for some aspects of aging, some aspects of cancer. This is an aging meeting. But since we published most of that last year, I hope to actually be able to show you some of the cancer data, because we find that some of the drugs we make are exceptionally effective against senescence-like cancer cells. So the goal for my lab was to find ways of eliminating senescence. This is for me a starting point. Actually, it's the 2011 equivalent of this experiment, but this is the one that wrapped it up nicely by Jan van Deursen, where he showed that if you genetically eliminate senescent cells from mice throughout life, there's a 25% lifespan extension. So by activating the suicide cassette, the attack suicide cassette, that's activatable by a drug that you can add to the drinking water of the mice. So only the senescent cells produce this suicide cassette. It's inactive unless you add the AP drug in the drinking water. This is the normal lifespan of mice. But now if you add throughout life, remove these senescent cells, you get a 25% lifespan extension. I think that was quite cool. The exciting thing is also that the maximum lifespan extension is not so much, so it's all median lifespan extension. But the real cool thing is that on the majority of parameters that they measured, they found that there was a health span extension actually. So I think that is the most important of everything we do. So since we are in Brussels, I always think we need to do a better job in convincing the European Union that we need to invest in anti-aging therapies, and things like this should be very convincing to go to the parliament, because we spoke at the parliament just earlier this year and I have no clue about any of this. So we're doing a poor job in conveying this message. Please take it upon yourselves to actually make this happen. Okay, so there are some compounds that were found by other labs that can eliminate senescent cells. These were either natural compounds, for instance quercetin, or anti-cancer drugs such as dasatinib or navitoclax. So we took it upon ourselves to see, can we actually develop drugs that could do this? So not just by screening, but actually by understanding the biology of what maintains the viability of a senescent cell, can you then exploit that to eliminate them? And I have to introduce you a little bit into FOXO-P3 signaling, which is the core of what we study. And this pathway is up in senescent cells. It's not always up on the mRNA level, but senescent cells are usually damaged, and usually you find at the sites of damage, at the sites of persistent damage, you find these two proteins back. So this we can actually quite nicely see in the majority of senescent cells. And then to develop drugs to eliminate senescent cells based on this knowledge, again, I'm skipping a large part of the introduction because I just want to go into the data. I have to actually go back to what for me started all of this, which was already a decade ago. And this was actually done in cancer cells. So at the time we studied melanoma as an escaper of senescence. So moles are all senescent because of their oncogenic BRAF mutation. And metastatic melanoma cells, majority of them, 80%, 70%, have oncogenic BRAF mutations plus something else, usually loss of P16. So that causes them to divide again. If in those cells we re-express the FOXO4 protein, actually we got blue cells in assay beta-gal staining, and this is an assay to detect senescent cells. At the time we didn't know anything about lamin B1, so this was the only thing we could do at the time. So that was actually exciting. That suggests that FOXO4 can actually enforce senescence. This was a P21 mediated response, so also interesting with regard to the previous talks. Because if you overexpress BRAF, nothing much happens in normal cells. FOXO4 induces a little bit. Now if you co-express them, you get a synergistic effect. So actually, now we know that BRAF through junc kinase can actually phosphorylate FOXO4. FOXO4 actually then binds the promoter of P21, where P53 is also present, and they together activate P21. The opposite is also true. If you take cancer cells that are very P21 high and they have an oncogenic mutation and they just happily divide, if you now remove FOXO from those cells, the cells became tunnel positive. So effectively a marker of apoptosis. At that time we didn't have CRISPR-Cas, so it's all shRNA based. The take-home message of this one slide, which was actually also 5 years of research, is that FOXO can dictate cellular fate. Either in intermediate levels, the cells divide. At excessive levels, the cells arrest, usually through quiescence, but if there's an oncogenic mutation through senescence or damage, then there's also senescence. And if it's a stress cell, you remove FOXO, then the cells die. So FOXO4 is a pivot. And this is all p53 mediated. So in p53 knockout cells, none of this works. That led us to the following model. It can be that in a situation of damage, and virtually most of the types of damage we've tested, we found this to be true. So oncogenic mutations that can lead to excessive growth factor signaling leads to stress, reactive oxygen species, or redox stress, or actual DNA damage. Then you get a binding between those two proteins, FOXO4 and p53. They hop to the p21 promoter, and you get senescence and the whole shebang in terms of interleukin secretion, maybe C-gas mediated. Actually, that would be interesting to find out. If you remove the FOXO4 protein, or as I will show you in a minute, you break the binding of the two, then you actually get a switch from senescence to apoptosis. So this is the take-home message on the mechanism. I will now go into a lot of molecular biology, and then I'll show you what we can do with such drugs. The problem is that these are transcription factors. So in 2008, everybody says, nice paper, dead end. Transcription factors are by default unstructured. Only the DNA binding domain of transcription factors has a structure, and you cannot drug transcription factors. So we went for kinases upstream, Jun kinase, ATM, HIPK2. None of that really worked. So I decided to actually block the binding using peptides. To do that, we also have to look a little bit at where FOXO is located in the cell. So here you see the DNA of a senescent cell, a human fibroblast in this case. And DNA-damaged senescent cells can develop what's called DNA scars. So these are basically glue on the sites of DNA damage. So the purple and the red proteins, they protect the open sites of the double-stranded breaks, and that prevents the cells from dying. And in those clusters, we find this FOXO4 protein to be present. So in green, FOXO4 is in PML bodies, and it's right next to 53BP1, where p53 is located. You cannot stain for p53 because they would stain the whole nucleus, but we know that active p53 is in those 53BP1 bodies. That's why we used 53BP1 in this experiment. Okay, so that's interesting. We find FOXO4, p53 present in DNA-damaged senescent cells. How do you break them apart? So I made a so-called D-retro-inversed FOXO4 peptide. We got it into the cells by using TOT from HIV. So HIV can infect human cells because of a very positively charged stretch. And here actually you see the evidence that what we developed was able to break the FOXO4-p53 interaction. Here we labeled FOXO4, isotope labeled it, and we titrated in increasing amounts of p53. And then you see two amino acids in FOXO4 that shift, these two. So this is the ground state, and when the p53 is added, you see a shift in these amino acids. So there's two basically amino acids that bind. If we then do the same experiment so again, this is labeled FOXO4 in red with a 1 equivalent of p53, and now we add 1 equivalent of the FOXO4 peptide, or 2 equivalents, you get the reverse. So basically this is the state when the two proteins are together, and now we add the peptide, and you see that actually you get a shift back to the ground state. So using this in vitro technology, and we use this a lot now in the lab, we were able to show that the peptides that we make interfere with the binding of FOXO4 and p53. So can you then use that in cells? Well, we were lucky that we had antibodies against the HIV TOT peptide stretch, so we were actually able to see how long it takes before the cells take up this FOXO4 peptide. It's 4 hours. So in 4 hours, the cells actually have taken up the majority of the peptide. Now we also have FIT-C labeled peptides, and we say the same thing. If we then look at the DNA scars, so again, that is the glue on the damaged site of the DNA in the senescent cells. Senescent cells have those DNA scars. The FOXO4 D-retro-inverse peptide completely annihilates those DNA scars. The same is true for PML and other markers of scars. Then back to P21. Actually, there it's quite interesting to look at the promoter because there's two p53 binding sites that flank one FOXO site. And P21 is very high in the majority of senescent cells, not all, because of course P16 is the alternative. But if you look actually at FOXO4 treated cells, they all lose their P21. So that's interesting because this is the glue on the DNA that protects the open DNAs from being fragmented. This is a pro-survival protein. It's an arrest protein, but P21 is also pro-survival. The third thing we observed is if you give senescent cells this FOXO4 G-retro-inverse peptide, you see actually active p53 excluded from the nucleus. So there's 2 sites in p53 that can become phosphorylated to activate p53, and then the serine 46 is pro-apoptotic. What you find in senescent cells, there is barely any signal. Now you give this FOXO4 peptide, massive amounts of active p53 outside of the nucleus. And this is actually all cytochrome C overlapping, or the majority at least, so that's mitochondria. So 3 things. This peptide unravels the glue that protects the open ends on DNA. It removes the Prosurvival protein, plus it actually kicks out active p53 to mitochondria, which is known to be pro-apoptotic. So on 3 levels, we have something that's pro-apoptotic with this drug. So to show that it works, now you can wake up, because this was all the molecular mechanism that I'm going to show. Actually, now we actually just use this drug in an assay to see if it can indeed selectively eliminate these senescent cells. So on the left, you see control cells. On the right, you see senescent cells. And they turn green when we add the drug. When they die, sorry. So and we add the drug. You can see that only the cells on the right become green. So irrespective of the mechanism that I just showed you, this is what you should remember. We have now developed a drug that basically can selectively eliminate senescent cells. So it was designed, it was not just repurposed. And we actually know that this is it targets the DNA damage specific subset of senescent cells only. So it's in that sense relatively clean compared to more general golden bullet anti-senescence drugs. So then can we employ this against aging? A favorite countryman of mine, of course, Rembrandt van Rijn. At that time, they had no clue what senescent cells were, but now we know. We have now developed a drug. Can we exploit then this knowledge to target mice that have aged? In our lab at the time, we made use of fast-aging mice. So these are mice that are based on human syndromes of humans that at the age of 10 look like they're already aged 80. So progeria, in this case, trichothiodystrophy. This mice, at a half year of age, is what it looks like. It has lost its hair, it crouches, has a lot of organ dysfunction. If you treat these mice with the FOXO4 peptide drug, 3 weeks later, the technicians came running to me and said, "What did you do with our mice?" So they actually regained their hair. So is this anti-aging? I don't know. It is encouraging at least. We were also able to change the behavior of the animals. So like old humans, old mice, they're quite immobile. So if you put them in a new cage, unlike young mice, they don't explore. Young mice, if you put them in a new cage, they start running around, they want to go away from us, they don't really like to be our friends. Old mice, they don't really care. They just sit in the corner. Here you see a small movie of 2 sisters, 2 aged TTD sisters. One of them was mock treated, and this one has received the FOXO4 drug. So actually, as you will be able to see, this mouse won't move throughout the entire movie, and this one starts running around again. And this we have tons of movies like this. So the interesting part is usually these mice, you can put them on your bench, go for a cup of coffee, and they're still there. The treated mice actually don't, so they actually explore again. So it's all very phenotypical, very descriptive, and I could have, of course, given you my best and worst example. So we also did actually analyses that Marco did, so running wheel behavior. Mice are nocturnal, they run at night when we are not around, so there's no way we can interfere with the experiment. And we saw actually that in those fast-aging mice, there's a 3-fold induction in running wheel capacity. So aging mice, they run about 1/10 of normal mice, and we could actually bring that up 3-4-fold. So that was very exciting and unbiased. So then we also looked at organ function. So in this case, we chose the kidney simply because we could find that in natural aging, but also in fast-aging TTD animals, there was a lot of urea in the blood. Urea should be in the urine, so that effectively means that the kidneys fail to function properly during aging. What we found is that the kidneys of fast-aging mice, they produce a lot of IL-6, so inflammation reminiscent of the SASP that was also introduced earlier, and they show loss of lamin B1, so the protein introduced in the previous talk. This is quantified over here. So fast-aging mice show a lot of IL-6, completely lost when you give the mice the FOXO4 drug. So you can actually wipe out the senescent cells quite nicely from the kidneys. If we then look at the kidney function, we looked at urea and creatinine, in this case in naturally aged mice. It is elevated compared to the young counterpart, and in both cases the drug could lower the blood values of this metabolite, suggesting that the kidneys function better again. So just to wrap up, as we age, we get more and more DNA damage. DNA damage can lead to senescent cells, and senescent cells secrete a lot of proteins. This can cause aging on all sorts of tissues, for instance, the kidney. And importantly, they produce FOXO4 P53 to shield that damage. If you now break that shield by a drug that we developed, so you remove the DNA scars, then those scars open up, the DNA opens up, and it's basically destroyed. On top of that, we have P53 going to the mitochondria, activating caspases and inducing apoptosis. So altogether, we were able to eliminate senescent cells using this drug and show a very good benefit on the health span of aging mice. And this is actually a restoration, so it's not just an extension of health span. So we were not able to do the lifespan experiment because I moved to another academic site earlier this year. But we did have actually a newspaper follow us for a year because they, of course, did not believe anything we said, as good newspapers do. So actually they were there again, like for 1 year. And March 31st this year, they published this article with a lot of nice graphics. And we actually had a low power experiment. And they actually followed us every month, sometimes even more times per month, to see whether we were lying or not. And actually they confirmed that the mice were healthier, and all of them were some of them were living more than 3 years of age. So that suggests that we can also confirm the genetic experiments from the Van Dersen lab. Is this then ready for human use? There are people who are doing that. I strongly advise against that, always, 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 because I think actually we need to optimize it further. At the moment, we see that the drug is about 10-fold selective for control cells, here in black, versus senescent cells. So this is a viability assay, incubated with increasing concentrations of our FOXO4 drug. And you see that it's about 10-fold more potent against the red line, so the senescent cells, than against the black line. Sounds very promising, but if you want to use enough that you get a good killing of the senescent cells, you are actually quite close to also hitting normal cells. And I'm really worried about people trying this on themselves because they randomly inject these drugs and they have really no clue what the buildup is in their body. So I think we should never do this. But it's unfortunately something which is already ongoing, and clinical trials are even started in Georgia now. So it's, I'm preaching to the wrong people, I think. So what we are doing actually to take this in a legal way forward, in a smart way, is that we founded a biotech company, disclaimer, disclaimer. This is one of the first times we have 3 people in the audience, and it was actually not intended by the board. So actually, Marco just presented earlier today, and Alexandra is in the back. And together, we actually started developing ways to improve the FOXO4 drugs. Here you see the structure of FOXO4, and you can nicely see it's like a hand. And together with P53, it's basically two hands holding each other. So this is what it looks like when it rotates. You see this alpha helix here. This is a very important one, and we started modifying that. Doing that, we actually now developed a drug which is 10 times more potent in inducing that shift on the NMR, which I introduced earlier. So we have actually a 10 times better binder than the first version. And then actually also on the biology, if you look at the amount of dying senescent cells that we can get by caspase activation, this was measured, it's actually also already way more potent. We are not entirely done optimizing this, but with this we want to go to the clinic. For the last 1 minute, sorry, I will actually talk a little bit on cancer. That's because, that's our new data, what makes the cell senescent? It's DNA damage, some types in the, at least. What we found is that chemotherapy-surviving cancer cells can become senescence-like. That suggests that our drugs might also be effective against therapy resistance in late-stage cancer. So just to prove that this is true, here we have actually the lethal dose of a few chemotherapeutic agents, including one cocktail. So it's a FAC, it's a mixture of 5-FU, adriamycin, doxorubicin that Marco talked about, and cyclophosphamide. And you can actually see that the parental cells have certain lethal doses to which they respond. But then what the cells that survived that treatment, the next round, they actually require almost double the concentration. And this is also what you see in patients. The first chemotherapeutic cocktail is the most effective. Thereafter, it becomes less and less potent. We found that the cells that survive that cocktail, but also the individual drugs, they all upregulate active P53 and FOXO in the PML bodies. So the cells that survive chemotherapy can become senescence-like by the DNA damage signaling pathway. And actually, if you quantify it for a larger amount of chemotherapeutic agents, we see that this is quite nicely true in general. They all have to be DNA damaging, though. Now, and then this is the last experiment, which I think is super exciting. If you now do look at what happens if you apply our FOXO4 drug to these cells, you can see that, for instance, by measuring apoptosis of breast cancer cells, the cocktail induces a certain amount of cell death. That's great. The FOXO4 drug does not do anything to the parental cells. But if you now do the same thing in the cells that survive the first round of treatment, then you see that actually the FAC cocktail is less effective in the second round, as you would expect, but boom, our FOXO4 drug gives a wipeout of the therapy surviving cancer cells. So this is something which for Clera we are now actually studying quite actively. So we actually identified already a new drug that's at low dose, very effective in 3D cultures of tumor organoids. Utrecht is all about 3D tumor organoids. And this is at some point where we want to go to the clinic with initially, because I think it's still very difficult to go for aging. But of course, aging is the goal in the end. And with that, I would like to thank the following people. Started already more than 10 years ago, then continued in the US, then mouse work in Rotterdam, and now we're back in Utrecht again. Thank you.